But the questions asking about location, so try to make sure you, I would say, answer the question, make sure your answer answers the question. I was reading some quizzes and grading some quizzes for microbiology 2-1, which is non-majors microbiology for nursing and vet students. They kept telling me something that was true, but not answering the question. It's like if I asked you to tell me the color of a fire engine, you said they have hoses, and they fight fires, and they have a siren on top. You're describing a fire truck, but not telling me the information that I actually want. And so that's sometimes students fall into that trap of they just memorize something and don't really think about what the answer should be without taking a step back and taking a step back. So as a general rule, take a step back. That was completely unintentional on my part. I did not do this on purpose. I don't know how these things showed up in your answer sheet, because it's just supposed to be locations. Why would there be anything else other than location in there? But there was that. And some people got it right. If nobody got it right, then I could give everybody 2 points for me making a mistake. But enough of you got it right that I don't worry about it. It's really hot in Oklahoma, in case you care. Super hot. I don't know what the temperature was like here on Wednesday and Thursday, but it was very uncomfortable. And those grass hours that were that big. But sometimes you have a tendency. But one got in my car as we're driving to the funeral, and that was not fun. Have to get a giant grasshopper out of your car. I don't know why you do. Not to bring everybody down, but hopefully you used your Wednesday off to good benefit. Because next Wednesday's our test. Today is lecture day. We'll finish our material, and maybe we'll start new material. I was in my office making some changes to the next set of slides because, you know, why not? I haven't posted them yet. Let's make some changes. You're like, oh shit, 5 minutes to class starts. Okay, so there's one more thing about the human and chimpanzee chromosome story that we talked about or tried to talk about last Monday. It's not exactly about what makes the human chromosome and the chimpanzee chromosomes different. It's a study that was done in chimpanzees by themselves. And one of the things that this study found, which is also true for other studies of a similar fashion, with studying chimpanzees from different regions, they found that the difference between a Western chimpanzee and an Eastern chimpanzee is greater than the difference between a human and a chimpanzee. So think about that. 2 different groups of chimpanzees have more genetic variance than a human and a chimpanzee. What do you think that tells you about genetic variation? Wouldn't you expect 2 groups of chimpanzees to be closer to each other than another completely different species? So that's a little weird, that the difference between humans and chimps is smaller than the difference between 2 different chimpanzee groups. It was another study, and it's related to that. There was the study we talked about with skin color. They took different nations from Africa and looked at the African populations, and found that between one side of one country and the other side of another country, there's more genetic diversity within that country than if you compared that country to African Americans in the United States, for example. And so, in a small region, you can have a lot of genetic diversity. And the further you get away from each other, we might expect to be more genetic diversity based on distance, but that's not what we see when we actually get to the DNA sequence. Which maybe makes a little more sense. More genetic diversity in smaller areas is actually better. And as you get further and further away, you don't need to worry about genetic variation as much because there's not as much interbreeding between those populations. And really with sexual reproduction, the problem is when you have disorders and recessive traits causing diseases or a dominant trait getting diseases, you want to sort of try to keep those minimal in the population. But you could be more closely related to someone from Europe than you are from someone in East Tennessee, genetically speaking, if you look at the DNA sequences. And so distance really doesn't do much in some cases. Now, one of the other studies I want to talk about when it comes to molecular genetics is this one that was fairly recent. Here's the date. 2022, late October. This guy won a Nobel Prize, so he's a pretty big deal, the Nobel Prize. I can't remember which one it is, I think it's the guy with the ponytail. So he was doing an analysis of an early human that was found in a cave in Russia. And it's from the Denisovan region of Russia. Therefore, they call it a Denisovan human. This word here, Denisovan human. Turns out that Denisovan humans were around at the same time as Neanderthals. And also early humans, because Neanderthals and humans are different species. And what was interesting about this individual they found is it appears to be a hybrid. Half Neanderthal, half Denisovan, and half something else. So you know, it's a three halves, but that's sort of the idea. But you really only identify some of what is in this individual. And that's the power of DNA sequence analysis. So you can try to find these early hominids and trace our descendants from them, or try to look back in time to see, well, what maybe we look like back then to what we look like now. Now, the other thing that's sort of fun about these studies is they talk about how much Neanderthal DNA is still in human populations today. Does anyone try to guess? How much Neanderthal DNA do you think is in modern humans right now? Anyway, just go out a number, we'll see higher or lower.
30%.
It's lower than that.
3.
Typically higher, and they say about 5% of our DNA is from Neanderthal origins. Sometimes higher. There's one study that suggests that humans, only 8 to 10% of the human genome is uniquely human. Meaning some of our DNA, a lot of DNA came from early hominid species. Only 8% makes us the difference that we see with humans versus all these other things. That's kind of neat, looking at different things. This paper talks about that difference. Where's the little quote they say? We find that only 1.5 to 7% of the modern human genome is uniquely human, which means we're a amalgamation of all the different things that came before us, make us into what we are today. And this one study looked at different regions of Europe and Asia, and they found that depending on where you are from, you have more or less Neanderthal DNA. And so 2% of Neanderthal DNA in Eurasia, East Asia can be as high as 8%. So where we know Neanderthals are typically from, we expect more people from that same area have more Neanderthal DNA. People that are further away from where Neanderthals were probably should have less Neanderthal DNA. Now, the cool thing about molecular genetics is we get to DNA sequence things, which allows us to identify some interesting things. And one thing we like to look at are homologous genes. We're going to talk about homologous chromosomes quite a bit in this next section. A homolog is something that is similar, but not exactly the same. And generally, when we talk about genes and proteins being homologous, we typically mean they have the same DNA sequence or a similar DNA sequence, the same protein sequence, the same protein shape, and the same protein function. So let's get that linked together. DNA makes RNA, RNA makes protein. So any changes in DNA would affect the protein sequence and maybe the protein shape. And protein shape is directly related to protein function. That's what I found. I don't know why that makes me happy. It does for us. Let's come back. For a gene that makes a protein, and that gene is also found in other organisms that should make the same protein, we call these orthologs. How many of you have taken organic chemistry? Who loves ortho-paro meta bullshit? Nobody? Well, it's a sort of a similar thing. An ortholog is a homolog that's in a different species. So Orthologs should do the exact same thing. The enzyme that does alcohol dehydrogenase is the same in every organism that has alcohol dehydrogenase. One of the important enzymes in glycolysis is glucose 6 kinase. I forget what it's called. Doesn't matter. Let's call it kinase. Just for fun. But every organism that does glycolysis has that same gene, has that same protein. So these are the places that we can look for similarities. Orthologs. We all do the same things. We should all have the same genes because we all have the same proteins. And orthologs tell us an evolutionary relationship between people and things. Imagine the very first cell made its first genes, and that gene was the gene that was passed on to the next generation, and the next generation, until we get to everybody we see today. So that is present in everybody, or most people we call those orthologs, because they pass through sort of evolutionary history from parent to offspring and so forth, generationally speaking. There are rare events that will occur during a normal organism's lifetime where a gene gets duplicated. This ties directly into the idea of CNVs. Never copy number variation. It's a really big type of DNA duplication event or deletion event. But if a single gene gets copied, and you get 2 copies of the gene, now you have potential for genetic variation and evolution to occur. And if a gene is duplicated within an organism, meaning an organism has now 2 copies or multiple copies of the same gene, we don't call them orthologs anymore, we call them paralogs, because they're in the same organism. Ortholog, they're in different organisms. And the cool thing about paralogs is that extra copy can undergo differentiation, or it can undergo a process called inactivation. When you have an extra copy, you don't necessarily need it. And gene dosage is a problem that we need to deal with. So having extra copies is sometimes bad. So sometimes the extra copy is simply allowed to mutate to the point where it's no longer functional. If that happens, we call that event now a pseudogene. So if a gene duplicates itself, and that new copy or the old copy, doesn't matter, one of them encounters or accumulates enough mutation where it makes a protein that's now non-functional. It might still have a start codon and a stop codon and a promoter and a terminator, but it's no longer going to make a functional protein. We call that a pseudogene. There's a lot of examples of these pseudogenes in organisms. Basically kind of like evolutionary dead ends. You try to make evolution happen, but nothing happens, so it's just kind of dead, so wait. Kind of like vestigial limbs. You may know about vestigial limbs. Where we have tails, but we don't have tails. Snakes have legs, but they don't have legs. The remnants are there, the similarities, but it's no longer an actual tail, so our tail bone is really short, so like really long, like a monkey's or any other type of tail individual. But pseudogenes is when mutations occur to make it non-functional, but sometimes those mutations that are going to accumulate are going to give the new gene, therefore the new protein, different characteristics. Sometimes those characteristics just change the kinetic properties of the enzyme. If you've ever heard of Km and Vmax for enzymes, sometimes you can change how good or bad something is. What we really care about for evolution is sometimes these paralogs take on new functions. And so let's say the protein eats sugar A, and it does something to it. Let's say it adds a phosphate group to it. If you take the paralog and you mutate it enough, maybe instead of using sugar A, it uses now sugar B. But it does the same thing to it, so it adds a phosphate group to it. Now we could use sugar A and do something else with it. That could be the other way this thing can change. But paralogs allow for evolutionary events to happen where we can get genes with new functions or slight variations in functions. But a lot of times they simply go nowhere. It's not exactly the same thing, but the transposons that are in our genome right now have accumulated mutations over time that have inactivated them. We don't call them pseudogenes, because they're not genes, they're transposons, but maybe we could call them pseudotransposons, because they're still there, they kind of look like transposons, but they don't work anymore. And paralogs are really fun to think about. And think about, why do you have extra copies of the gene, and what do these extra copies do? And one example that we have in humans is the gene called HOX. And you don't need to memorize the number 39, but imagine having 39 different copies of this gene. They're located in 4 different chromosomes, so I would probably say that originally there was 4 copies. Because typically when you get gene duplication events, they stay in the same chromosome. So if we had 4 copies originally, that sort of did something, but now we have 39 of these. And if you care about what HOX does, it does some really cool things. I don't know, maybe if we had more of them, we'd have more limbs and more ears, or less ears and less limbs. Kind of fun to think about. I don't know what you say about it. Other than it happens. It's out there. The HOX specifically is an important gene because it does, these mutations in HOX can lead to certain types of cancer. You don't need to memorize any of these, it's just a fun illustration of things. But different HOX genes mutated, making different types of cancers. Which makes sense. HOX is involved in cell division, and one of the things that we talk about later on this semester is if you can break the process of cell division in some way, you can lead to a tumor cell and cancer. Just because I'm egotistic, I get to talk about my research as a grad student. I studied paralogs as a graduate student. And it's kind of a surprise, because we didn't know they were there. It's sometimes fun when you start off with something. And what I found was that they all had different kinetic properties. Yay. Paralogs can have different functions. They can use different substrates but do the same chemical reaction. They can do the same chemical reaction. I said same substrate, different chemical reaction, or different substrates. Wait, same substrate, different chemical reaction. Different substrate, same chemical reaction. Actually, you memorize that. That's sort of the thing you want to think about. The last bit of fun that we'll encounter for exam 1 will be a calculation. It's a very simple calculation on the surface. It's a allele frequency calculation. Not to be confused with penetrance, because we do have 2 different calculations to deal with. The most common problem that arises because of or from the allele frequency calculation is people forget diploid exists. And when we look at how many alleles are there, everybody has 2 of each. So a lot of people will do math wrong. If you don't do the math right, you don't get the right answer. And so you always have to consider, it's always about the number of individuals that are present. But you have to remember each individual is diploid, therefore there are 2 of every allele. We also have to understand that homozygous and heterozygous represent different allelic meanings. This is the other place where it can make a mistake. That's the least common mistake that's made. By far, if you get this calculation wrong, it's because you divide by the number of individuals rather than by the number of alleles in the population. If you pay attention, you'll notice your mistake. Now, when you divide by the number of individuals, you get a number that's pretty high. You might get 90%, or sometimes even 100% or more, and that should never happen, because the alleles should have to be 100 even. So in this case, we'll take a simple case. 795 homozygous dominant, 190 heterozygous, and 15 homozygous. So what is the frequency of the dominant allele, and what is the frequency of the recessive allele? And it's on the next slide, but hopefully you can think about how we do this. Homozygous means 2 copies of the same thing. Heterozygous means 1 copy. of two different things. So if we're trying to calculate the frequency of the dominant allele, we take 190 times 1 plus 795 times 2. And again, divide that by the total number of alleles in the population, which is the number of individuals times 2. Here we get 89%, and which means by extension, the other thing has to be 11%. That can also help you with the math. You don't have to do both calculations. You can simply do one calculation and then do another calculation, subtract the frequency from the total. It is a frequency, so it's kind of like a probability, so it's either a percentage or leave it as a decimal number. Don't just write 89, that's wrong. It has to be 89%. That's easy, there's only 2 alleles to deal with. If you looked ahead, you probably know what's coming. You have cases where there are more than one allele that we have to deal with. And I always use blood type as a good thing because blood type is fun to think about. There's a lot of varieties in genotypes, which means in this one question, there are 3 different calculations that can be performed. So we'll start with the simple case of looking at the frequency of the A. And the next slide will walk through this. But how many of these we need to use if we're going to calculate the frequency of A? If there are 3, this one has an A, this one has an A, this one has an A. So we ignore these numbers, ignore that number, because they don't have the allele that we're interested in. So we take 14, 6, and 1. We take 14 times 2 because that's a homozygous situation. The other 2 are heterozygous, so we can just take them as our R, or follow the pattern which is multiplied by 1. So 28 plus 6 plus 1. And we have to divide by the total. Luckily for you, I made the total already. We take 28 plus 6 plus 1, that's 35, and divide by 94, which is the total, and 37.2. And the question is, how many of you are going to forget to take the total number of alleles? If you just added up 21, 14, 6, 2, 3, and 1, that's
47.
47. You divide 35 by 47, you get something like 90 something. That would mean the other alleles would be pretty small because all add to 100. So keep that in mind. A lot of people are just going to blindly do the calculation, not think about it. But if you kind of know what you expect, if there's 3, in this case there's 4 alleles, well, 3 alleles. So if one is 90, there's not a whole lot of space for the other ones to be there. And the one that is the most is the lowercase I. So we might hopefully notice that it is weird. The thing that has the most is not going to be the most percentage if I look at it that way. So try to pay attention to what you're calculating, and you're less likely to make mistakes. And as a general rule, I like 3. I can't call them significant digits because that's a chemistry thing, but I like 3 digits in my answers. Sometimes I say 3 non-zero digits, that's sort of throws people off. Sometimes it might be like 307. I think they need to put another one in there. There's a big difference between 37.9 and 37.2. This is why I need that extra little digit at the end to make sure you actually did it right. So just to see if we can do it correctly, which means would you like to do in class now? B or I or O? There's one vote for B. Nobody else voted, therefore we will do B. What is the frequency of B? Who can beat me to the answer? Don't look at me. Look at your own face paper. Don't make fun of my southern accent either. What? What'd you get? It was 94, yeah.
Same as the last time.
Yes. I know I wrote 95. Let's do some longer. Okay, no, that's not right. November. No, that's right. 1. I know it's backwards. B. And backwards. What's the answer? Hold on. Let me get my calculator. I got two different answers here. So
You didn't, it's not double. So it should be an 8 over 94.
can't do that.
That's what it should. 8.5.
8. Are you sure? 8.5? Is that what you want to go with?
8. Wait.
No,
It's
it's
0.85.
Is that what you want to go with?
Oh, sorry.
Is that what you want to go with? Is that what you want to go with? You want to just do two numbers here? That's what two numbers you want to do, right?
0.085.
What? Oh, 8.5?
Yes.
1. So it's 8.5%. So if that's 8.5% and the other one was 37.4%, how much is I? Well, it's 37 plus 8 plus a 1, and the rest of it should be the I. But that's how you do allele frequency calculation. Just don't forget to divide by the total number of alleles. It's very easy to divide by the total number of individuals. But luckily for us, on exam 1, it's never correct to divide by the total number of individuals. For penetrance, we don't divide by the total number of individuals. And for allele frequency, we don't divide by the total number of individuals. This does end the information we need for exam number 1. Would anybody like to ask any questions right now about exam number 1? Because a lot of time left, so we're going to go ahead and fill your head with the fun esteem for students.
Don't remember.
Material that won't be on the exam before the exam starts. Yes.
And did you have it posted these days?
I have not posted these yet. I know, such a dastardly dude. But it's just meiosis. Everybody knows about meiosis. Well, maybe you know about urosis. Or horosis. Or hisosis. Or therosis. Or usosis. Nobody likes puns. I mean, pronoun puns are really fun puns. It's not arosis. Urosis. I guess it was urosis, although it's ulosis. Anyways, now the tie in between what we've been talking about and meiosis is a little bit about genetic variation. And the importance of mutations are going to keep sort of following along. We're going to do this cell division process, meiosis. It'll lead into talking about replication and a few other fun things about DNA changes. We'll talk about meiosis. Mitosis. Later on, that'll be an exam 3 things, so break up the cell division for various aspects. It does make things a little weird because we're going to talk about the steps of meiosis and then ask you to remember the steps of meiosis when we talk about mitosis, and then later on for the exam final, so do some comparison speakings. But hopefully, a lot of you remember a little bit about what happens during meiosis. And what is mostly important about meiosis is its place in the process of sexual reproduction. A lot of people will sometimes mistakenly state that meiosis is the same thing as sexual reproduction, so we do want to make sure we understand that sex does take 2 steps. Meiosis and then the fertilization step. And both are going to provide places for genetic variation or introduce genetic variation. But we're not going to worry about the fertilization step right now. We will when we start doing some Punnett square analysis later on, which will come up directly after this, so that'll be fun. When it comes to the actual phase of meiosis, there are some important genetic events that will occur. And we're going to have to go back to Gregor Mendel and look at his principles of inheritance to think about what's going on, how these things relate to each other. So as a sort of a fun little funness, this section will look backwards for exam 1 and also look forwards to exam 3. But right now we're not going to focus on fertilization. We're going to focus in on meiosis. And one of the important things about any type of organism undergoing or using the sexual reproduction process is how you maintain chromosome number from parent to offspring. You can't just use the cells that you normally have and give them to your offspring because that's not sexual reproduction. By definition, sex requires DNA from different individuals to come together to fuse to make the new offspring. And that's fertilization happens when those two things coming together creates a significant amount of genetic diversity, but you're combining DNA from two different sources. But how do you make the DNA that you need to give to the next line of individuals? That's where meiosis comes into play. So for diploid organisms, we have a diploid state and then a haploid state. The haploid state will go back to the diploid state eventually, and then the process will continue on to this mostly diploid state or a little bit of the haploid state. Now, we only look at humans because I don't care about plants. So if you know anything about alternation of generations and the funness that is diploid to haploid to haploid to diploid to diploid back to haploid, whatever it is, I don't know. We don't care. We're just trying to understand the genetic variation aspects of meiosis. We're not going to spend a whole lot of time on trying to understand the steps themselves. Cell biology will deal with the steps in a little more detail. I'm just going to focus in on the genetic variation events for the most part. We need to understand that we have certain types of cells that are going to perform the process of meiosis. We call them germ cells. Sometimes you could also call them sex cells. So ovaries, testes are the cells that are in our body that are going to perform the process of meiosis. Liver, heart, lung, skin, these are all going to perform mitosis rather than meiosis. So meiosis takes a diploid cell and creates a haploid cell from it, or multiple haploid cells. Now we're going to use these haploid cells in the next step. So we take half of our DNA, or take all of our DNA and split it into halves. And then give half to the next generation, therefore the other half comes from a separate parent. And by going through the process of meiosis, we are going to introduce genetic variation. It's going to happen in two different ways. One will affect a single chromosome, the other is going to affect multiple chromosomes as we sort of look forward. I like to call it an intracellular or intrachromosomal change versus an interchromosomal difference. Intramural sports versus intramural sports, for example. And you know, intramural sports? That's when UTM goes and plays Georgia or Tennessee or anybody else from the OEC. And if frat A plays frat B, that's what's called intramural. Within the organization. Anyways, intra versus intra. Now again, with the meiosis step, making a haploid cell, we take those haploid cells through the process of fertilization, combine two together to remake a diploid cell that's now called a zygote. So we now have maintained the chromosome number from generation to generation. Imagine how much weirdness would happen if meiosis either didn't happen or didn't always happen. What if it wasn't just haploid cells that fuse together? Because this happens with plants. You get allopolyploidism and autopolyploidism. So we get organisms that have a significantly large number of chromosomes. So we have 46 chromosomes. Imagine if we just didn't do meiosis first. And we fuse two cells together. How many chromosomes do we have? 192. that 192 person now, instead of reading to a 192 person, reads to a 46 person? How much do you need that person to have?
That's 46,
I don't know, you said one,
So I said 92.
you said 192, I heard you. Well, fine, let's do 92 plus 92, and then we'll get to 184. Take that. If this goes on, then what if we throw in a meiosis event in there? So maybe there's a 23 in there somewhere. Now it's 46 plus 23, or 92 plus 23, or 184 plus now 23. Or who knows what happens? It's the Wild West of how much DNA you have. Who knows what can happen if eventually too much DNA, not enough nucleus is sort of the issue. Again, we will deal with the random fertilization event idea later, but it will be when we deal with Punnett squares. But right now, again, just want to focus in on the first aspect, which is meiosis. And just for fun, this slide will be in your set of slides, just so you can lament that we're not going to talk about this at all. The stupid plant. I know, alternations and generations is kind of cool. But we're humans, we only care about animals. Something that is both exam 1 and exam 2 is this idea of diploid versus haploid. So no one should get this wrong because we're now reviewing important terms for exam 1 and exam 2. Who's going to forget what diploid means? Anybody? Hopefully not. 2 copies of each chromosome. 2 different copies, because this is sexual reproduction. Half your DNA comes from one parent, half your DNA comes from the other parent. And because these DNA molecules are from different origins, we can't call them the same, we have to call them similar but different. We use the idea of homologous chromosomes. And we talked a little bit about what makes homologous chromosomes same but different. That'll be on the next slide to remind you. We do need to understand that our DNA is slightly different from chromosome 1 to chromosome 1. And as we go forward through the process of meiosis, going from a diploid state of having a maternal one and a paternal one, we go to a haploid state where we have just one or the other. If we half our DNA, so we lose some portion of the maternal or paternal idea or essence. It's not lost, it's in a different cell. Because we're not losing DNA, we're just putting half of our DNA in one cell, half the DNA in the other cell. And this is a random process. It's not just all dad's DNA in one cell, all mom's DNA in another cell. It's a combination of, you know, maybe 50-50, 70-30, could be 90-10, who knows? There's some proportion of chromosomes in each different type of cell. But because the homologous chromosomes do come from different parents, we can almost guarantee that the DNA sequence of the maternal chromosome 1 will be different than the DNA sequence from paternal chromosome 1. They're going to be very similar. And when we talk about homologous chromosomes, same gene order, same gene content, same length, same centromer position. It's the same chromosome. It just came from a different origin, so it's going to have a different DNA sequence. And last time we talked about the alleles being different when we compare homologous chromosomes to each other. And that could be technically true, but also could be technically false. Because you could have the recessive allele in both of your chromosomes, but it could be a different recessive allele. Because sometimes there are multiple recessive alleles in a population. Again, there's a lot of these single nucleotide polymorphisms that are out there. Some of them are not going to cause any variation whatsoever. So even if you do have the exact same allele, the DNA sequence is not always going to be 100% identical. So there's going to be some variation between mom's chromosome and dad's chromosome. Shouldn't have to tell you what a chromosome is. Just a piece of DNA. What do the chromosomes look like in eukaryotic organisms? They're linear, there's lots of them, typically speaking. which is not the case for prokaryotic organisms. There's only one chromosome. And on that last slide, you might have, or a slide before, you might have noticed something about prokaryotic organisms. They only use haploid in relationship to diploid. You can't just be haploid and never be diploid. So for prokaryotic organisms, they're not haploid at all. They're just prokaryotic. Haploid only has meaning in the context of diploid state. Now, unfortunately, when we go through the process of meiosis, we are following a predetermined set of events, and that set of events is based off of the process of mitosis. In the process of mitosis, the first thing we do before we start the replication of, or duplication, or division of cells is to copy all of our DNA molecules. And so if you make an identical copy of a chromosome, we need a term to describe that now identical copy. We can't use homolog anymore because that's already been used. So the term chromatid was devised. And unfortunately, if you have poor enunciation like me sometimes, because I kind of mumble and talk fast, chromatid may sound like chromatin. Please make sure that you are good with the use of chromatid and the use of chromatin, like heterochromatin and euchromatin. But a chromatid is considered an identical copy of a chromosome. And when we copy a chromosome, the copy stays connected together with its template. So because of this, we call this a sister chromatid pair. As a general idea of what we're going to see happen as we go forward, take a case where you have homologous chromosomes. I'll just use different colors to represent two different things. If only. This is what we call a 2N state. And how many is N? Yeah, in this case, N is 1. Let's say N is 1. For simplicity. Because there's only one chromosome 1, and there's 2 copies of it, which makes it diploid. But when we go through the first step of any cell division, we make copies of our chromosomes. And generally speaking, when we draw sister chromatin pairs, we draw them like this. So we have one chromosome, that is now 2 copies. And this is the other thing that will be sort of hard to deal with for some people is when we talk about DNA, this is still one chromosome, but it is 2 DNA molecules. And each DNA molecule is a double-stranded DNA molecule, so some people get sort of confused with what is DNA and what are we talking about. This one chromosome becomes one chromatid pair, which means it's 2 individual chromatids. We have, again, 2 DNA molecules. One chromosome, 2 copies of that one chromosome. And so the way I like to think about it is now this is no longer just a 2N state. And the way I like to think about it is now a 2N state. We're still diploid. So the 2N means we're diploid. The 2 in front of it means now we've duplicated all of our DNA, so we have 2 copies of everything. And some people like to use other things. I know Dr. Smart likes to put like a little superscript C or something on things. They're called chromatids or chromosomes. You've read in 140 lab. At a certain point, you might have seen that type of nomenclature. I just use this idea. It is important to understand how many DNA molecules are present and what are the DNA molecules that are present. And can we effectively talk about each one using the proper terminology. Now, as far as Monday goes, Monday is a day to ask questions, but if there are no questions or people just looking through the notes trying to waste time, I will lecture more on these slides that we posted to Canvas soon. In which case, we'll start with a fun little DNA exercise of how much DNA is there. Did anyone ask any questions before the weekend? You have 2 more minutes of class.
So this might be the question, but in that situation, with that right there, since it's a 2, 2 in, does that mean the n equals 2, or is it still equals
The n is still 1. So if you do the math, 2 times, well, you go 2 times 1, that's 2. 2 times 2 is 4. 4 tells you there are 4 pieces of DNA that are present there. But I don't like to call it 4n, because 4n could actually mean polyploidyism. And I want to separate polyploidyism from copying on a chromosome, which is not the same thing as getting extra copies of a chromosome. So I like this thing. So if you ever see 4n, I don't like that unless it actually is truly being used as a